Chapter 10 deals with photosynthesis. This is the process that converts solar energy, or energy from the sun, into chemical energy, and this occurs in organelles called chloroplasts. Autotrophs, also called producers, make organic molecules from carbon dioxide and other inorganic molecules. Plants are what we call photoautotrophs. They use the energy of sunlight to make organic molecules. Some organisms can use things other than the sun. They could use certain chemicals, but we're not going to be talking about those for right now. So A, B, C, and D show some examples of photoautotrophs. We have plants, we have kelp, we have some unicellular eukaryotes, and cyanobacteria. In this chapter, we're going to focus on plants. Some organisms, those that are called heterotrophs, obtain organic material by eating other organisms. They're also called consumers. They cannot make their own food. Some heterotrophs eat other living organisms. Others that are called decomposers decompose and consume dead organic material or feces. An example of a decomposer is a mushroom, which is a type of fungi. Almost all heterotrophs, including humans, depend on photoautotrophs for food and for oxygen. Chloroplasts, which are the structures where photosynthesis occurs, are structurally similar to and evolved from photosynthetic bacteria. While all green parts of a plant have chloroplasts, the majority of photosynthesis occurs in the leaves. Chloroplasts are found mainly in cells of the mesophyll. Each mesophyll cell contains between 30 and 40 chloroplasts. Now we're going to see during photosynthesis, carbon dioxide and oxygen are important in one way or another. Carbon dioxide enters and oxygen exits the leaf through microscopic pores on the leaves called stomata. Water is also important in the process. Water is absorbed by roots and travels to leaves through veins. Now, a chloroplast has an envelope of two membranes. These surround a dense fluid we talked about previously called stroma. There's a third membrane system, which are a bunch of connected sacs. Individually, these sacs are called thylakoids. When these thylakoids are stacked up in columns, they're called grana. Chlorophyll is the pigment that gives leaves their green color. Chlorophyll is found in the thylakoid membrane of the chloroplast. We're going to see later that chlorophyll is what absorbs light energy and drives photosynthesis. Figure 10.4 shows the location of photosynthesis in a plant. So on the right-hand side up top, we see the cross-section of a leaf. We see chloroplasts and the veins in the mesophyll. Then we see the stomata. Stomata, again, are where carbon dioxide enters and oxygen exits. Down below that, we see the structure of a chloroplast. Two membranes surrounding a dense fluid called stroma. And inside that fluid are stacks of thylakoids called grana. Photosynthesis is a complex series of reactions that can be summarized with the following equation. Light energy plus 6 molecules of carbon dioxide plus 6 molecules of water produces 1 molecule of glucose and 6 molecules of oxygen. Again, there's different sugars that can be produced, but we're going to focus on glucose, like we did when we talked about cellular respiration. The carbon dioxide is the substance that's becoming reduced, and the water is what's becoming oxidized. So what's happening in chloroplasts? They're going to split water into hydrogen and oxygen. They're going to incorporate the electrons of hydrogen into sugar molecules, and oxygen is going to be released as a byproduct. So it's going to basically reverse the direction of electron flow compared to cellular respiration. Now, while the products and the reactants are just switched, So just turn the arrow around. Basically, you have basically the same formula. The process is quite different. This is an endergonic process. If you see, one of the reactants is energy. Energy needs to be put in. We're going to get this energy boost by light. Photosynthesis consists of light reactions, this is the photo part, and the Calvin cycle, the synthesis part. The light reactions occur on or in the thylakoid membranes. During the light reactions, we split water, release oxygen, reduce the electron acceptor NADP+ to NADPH, and we're going to make ATP from ADP by phosphorylation. The Calvin cycle is occurring in the stroma. This forms sugars from carbon dioxide using ATP and NADPH that was generated during the light reactions. It involves carbon fixation, which is incorporating carbon dioxide into organic molecules, and reduction of fixed carbon into a carbohydrate. Figure 10.6 is an overview of photosynthesis showing the interrelationship between the light reactions and the Calvin cycle. You can see that during the light reactions, which are happening in the thylakoid membrane, water is used, the energy from light is used, oxygen is going to be released, and ATP and NADPH are going to be generated. The ATP and NADPH are going to be used by the Calvin cycle to turn carbon dioxide into a sugar. This is happening in the stroma. NADP+, ADP, and inorganic phosphate are going to be produced that can then be used in the light reactions. Before we talk about photosynthesis anymore, we need to know a little bit about light. So light is electromagnetic energy, also called electromagnetic radiation. It travels in rhythmic waves. The distance between crests of electromagnetic waves is what we call wavelength. The electromagnetic spectrum is the entire range of electromagnetic energy. Visible light is light that we can see. The wavelength varies between 380 nanometers to 750 nanometers. And we're going to see that the shorter the wavelength, the more energy something has. Sometimes light can behave as though it consists of discrete particles called photons. Figure 10.7 shows the electromagnetic spectrum. Visible light, again, is in the range from 380 nanometers, purple, to 750 nanometers, red. And again, the shorter the wavelength, the higher the energy. Up top, it lists different types of waves in the electromagnetic spectrum. So you can see some have more energy than visible light, and these are all harmful to humans. So there's UV light, which is from the sun. This can cause cancer. X-rays have more energy. When an X-ray technician takes an X-ray, they'll have a protective vest on, and they often go behind a shield or a window to take an X-ray. And then gamma rays, which are harmful. Other types have less energy than visible light, so infrared light, microwaves, and radio waves. Now, pigments are substances that can absorb visible light. Different pigments absorb different wavelengths. The wavelengths that they absorb disappear, so you don't see that color. Wavelengths that are not absorbed are either reflected back to your eyes or transmitted through the substance. Now, this is sometimes a hard concept to grasp, but leaves appear green, not because green is what's being absorbed, but it's because chlorophyll reflects and transmits the green light. So it's not using the green light. That's why it appears green. There are three important types of pigments in chloroplasts that are involved in photosynthesis. One is chlorophyll A. This is going to be the key light-capturing pigment. The second is chlorophyll B. This is an accessory pigment. This is going to transfer energy to chlorophyll A. And the third type are carotenoids. This is a separate group of accessory pigments. These can play a role in photosynthesis and some function in what we call photoprotection. So it absorbs excessive light that would otherwise damage the chlorophyll. Figure 1010A shows what wavelengths are absorbed by these different pigments. So chlorophyll A has some key absorption in the violet blue range and also in the red range. So that's why it appears as a greenish color. Chlorophyll B absorbs best in the blue range and the orange-red range, so it appears as an olive green color. It doesn't absorb that. Carotenoids do best in a blue and a purple color, and they appear orange. 1010B shows an action spectrum, and this shows the relative effectiveness of different wavelengths of radiation driving a process. So this is showing, in this example, the rate of photosynthesis, and this was measured by the amount of oxygen released, because during photosynthesis, oxygen is released. So the more oxygen released, the more photosynthesis is occurring. And you can see that around about 430 and around maybe 680, those are the wavelengths that are most effective in driving photosynthesis. When a pigment molecule absorbs light, one of its electrons goes from a ground state to an excited state. It has more potential energy in this excited state and is less stable. It's quickly going to fall back to the ground state, where excess energy is released as heat. In thylakoid membranes, chlorophyll molecules are organized with proteins into complexes called photosystems. They have two parts. There's a reaction center complex that's surrounded by several light harvesting complexes. The light harvesting complex consists of chlorophyll A, chlorophyll B, and carotenoid molecules bound to proteins. The light harvesting complex is responsible for capturing light energy. When the light hits a pigment molecule, one of its electrons is going to be raised from the ground state to the excited state. Energy is going to be released as it falls back down to the ground state. This energy is going to be transferred from molecule to molecule and eventually funneled to the reaction center complex. The reaction center complex is an association of proteins holding a special pair of chlorophyll A molecules and a primary electron acceptor. The pair of chlorophyll A molecules can use the energy from the light harvesting complex, boost one of their electrons to a higher energy level, and transfer it to a primary electron acceptor. Figure 1013 shows the structure and function of a photosystem. Purple shows the light harvesting complexes, and the blue shows the reaction center complex. A photon of light hits one of the pigment molecules. Electrons are elevated to an excited state and then go back to a ground state, and energy is transferred from one pigment molecule to the next, eventually being funneled to the special pair of chlorophyll A molecules. One of their electrons gets boosted in energy, and this travels to the primary electron acceptor. On the right-hand side is just showing a picture of a photosystem using X-ray crystallography. There are two types of photosystems in the thylakoid membrane. Photosystem 2, PS2, functions first. The reaction center chlorophyll A of PS2 is called P680. This is because it is best at absorbing wavelengths at 680 nanometers, which is the color red. Photosystem 1, PS1, functions second. Its reaction center chlorophyll A is called P700 because it is best at absorbing wavelengths at 700 nanometers. This is the far red portion of the spectrum. Photosystem 1 is called Photosystem 1 because it was discovered first, even though it actually functions second in this process. During the light reactions, there are two possible routes for electron flow, cyclic and linear. Linear electron flow is the primary pathway and what we're going to focus on. It involves both photosystems and produces ATP, NADPH, and oxygen using the energy of light. Cyclic electron flow uses only photosystem 1. Some organisms only have photosystem 1 and only produces ATP. Now, in linear electron flow, as electrons flow down an electron transport chain from photosystem 2 to photosystem 1, energy is released during redox reactions and is used to create a hydrogen ion gradient. This is going to pump hydrogen ions to a higher concentration, not the way they would want to go, into the thylakoid space. They're then going to want to come back over, like we talked about when we talked about cellular respiration, and this is how ATP is going to be produced. The electrons entering PS2 come from the splitting of water. Chloroplasts and mitochondria we talked about in the last chapter when we talked about cellular respiration, they both generate ATP by chemiosmosis, but they use different sources of energy. So mitochondria transfer chemical energy from food to ATP. Chloroplasts transform light energy into chemical energy of ATP. In mitochondria, protons are pumped to the inner membrane space and drive ATP synthesis as they diffuse back into the mitochondrial matrix. In chloroplasts, protons are pumped into the thylakoid space and drive ATP synthesis as they diffuse back into the stroma. Figure 1017 just kind of shows figuratively what I was talking about in the previous slide. So on the left-hand side, it has the name for the different parts in the mitochondria, and on the right-hand side, where it's happening in the chloroplast, you can see they both involve an electron transport chain. They both involve the enzyme complex ATP synthase, and in both, ATP is going to be made from a proton gradient. The difference is where the different processes are happening. This would be a good slide to understand for the exam. ATP and NADPH are produced on the side facing the stroma, where the Calvin cycle takes place. In summary, light reactions generate ATP and increase the potential energy of electrons by moving them from water to NADPH. Now this diagram is a little more complex than what we talked about. I tried to make it as simple as possible, but here are some of the key things as an overview. You will need to know slightly more details, but those I've already gone over. So what's happening here? Light is hitting a pigment molecule in the light harvesting complex of photosystem 2. Water is being split. Energy is being funneled to the reaction center pair of chlorophyll A's, and one of its electrons is going to the primary electron acceptor. Oxygen is released when the water is split. The electrons travel down an electron transport chain. Light is going to hit photosystem 1. And we have two things being produced. One, we get ATP produced. That's coming from the photosystem 2 in the hydrogen ion gradient. Another thing that's happening is we're getting NADPH produced. And this is happening from the electrons traveling down the electron transport chain that are coming from photosystem 1. NADPH and ATP are then going to be fed into the Calvin cycle. So we talked about the left-hand portion of this process in this slide, the light reactions. Now we're going to talk about the Calvin cycle, where the ATP and NADPH generated from the light reactions are going to be used during the Calvin cycle along with carbon dioxide to produce a sugar. The Calvin cycle is going to use the chemical energy of ATP and NADPH to reduce carbon dioxide to a sugar. This is an anabolic process. It's building sugar from a smaller molecule. So where does the carbon come from? Carbon enters the cycle as carbon dioxide. It's going to leave as a sugar named glyceraldehyde 3-phosphate, G3P. In order to get one molecule of G3P made, the Calvin cycle has to take place 3 times. So basically, we're fixing 3 molecules of carbon dioxide. The Calvin cycle has 3 phases to it. There's carbon fixation, reduction, and regeneration of a 5-carbon sugar called RUBP. So figure 1019 shows the different steps of the Calvin cycle, and I'm going to tell you the important things that you need to know. So one, you should know that 3 carbon dioxide are going to enter for each cycle. If you look at the yellow stars, they show the ATP that is being used. On the right-hand side is 6. On the left-hand side, you see 3. So 9 ATP are used during the Calvin cycle. In the little orange box on the right-hand side, you see NADPH. 6 NADPH is going to be used during the Calvin cycle. So the Calvin cycle consumes 9 ATP and 6 NADPH. These came from those light reactions we just went over. And we're going to produce 1 G3P. Now, the one step that I actually want you to know is the first step. So in the first step, we have carbon dioxide being added to RUBP. RUBP stands for ribulose biphosphate. It's okay if you just know it as RUBP. In order for these to combine, it needs the help of an enzyme. The enzyme is what's in the little bluish-purple box, rubisco. So carbon dioxide combines with RUBP with the help of rubisco. It's going to make a 3-carbon molecule called 3-phosphoglycerate. The reason why this step is important, and I want you to know it, is because 3-phosphoglycerate, like I said, it has 3 carbons. If you look, there's 3 little blue balls. This is important because the Calvin cycle is sometimes called the C3 cycle. And it's called C3 because the first intermediate formed has 3 carbons. We're going to look at some alternative methods, and we're going to see that they have different names because there's a different intermediate formed. But for right now, I want you to understand that first step. I want you to know how many ATP and NADPHs are used, and that our main output is G3P. If you look on the left upper hand side, you'll see that during this cycle, RUBP is being made again. That's so it can then be used for the next group of carbon dioxides. Alternative mechanisms of carbon fixation have evolved in hot, arid climates. Dehydration is a problem for plants. Plants lose water through stomata. So on hot, dry days, plants close stomata. This conserves water, but it limits the ability to do photosynthesis. That's because when stomata are closed, it reduces access to carbon dioxide because that's how they normally enter. At the same time, oxygen is going to build up because normally it exits through stomata. So there's going to be a buildup of oxygen. So if there's a buildup of oxygen, this is going to favor a wasteful process called photorespiration. So what happens is that Rubisco, the enzyme we talked about in the previous slide, it's going to add oxygen instead of carbon dioxide in the Calvin cycle. This is going to produce a 2-carbon compound, not that 3-carbon compound that we talked about. So photorespiration consumes oxygen and organic fuel and releases carbon dioxide, but it's not going to produce ATP or sugar. So this is definitely a bad thing. We don't get any ATP, we don't get any sugar, so this is a process we don't want to happen. Photorespiration is believed to be an evolutionary relic because Rubisco first evolved when the atmosphere had far less oxygen and more carbon dioxide. In many plants, photorespiration is a problem because on a hot, dry day, it can drain as much as 50% of the carbon fixed by the Calvin cycle. One alternative is what C4 plants do. So they minimize the cost of photorespiration. Before the Calvin cycle, they incorporate carbon dioxide into 4-carbon compounds. So they're going to separate where carbon fixation occurs, which is in the mesophyll cells, and where the Calvin cycle occurs. In C4 plants, the Calvin cycle is going to occur in cells called bundle sheath cells, which is different than what we talked about before. In C3 plants, everything was happening in mesophyll cells. So they're going to separate where these two processes occur. These bundle sheath cells are arranged in tightly packed sheets around the veins of the leaf. Mesophyll cells are more loosely packed, and they're between the bundle sheath cells and the leaf surface. So sugar production in C4 plants is a 3-step process. So you're going to notice here that we're using a different enzyme than we used before. The enzyme that's going to be used is called PET carboxylase. This is going to add carbon dioxide to a molecule called PEP. This is going to produce OAA in the mesophyll cells, oxaloacetate. The reason they use PET carboxylase is because it has a higher affinity for carbon dioxide than Rubisco. So even if the carbon dioxide concentrations are low, it's still going to fix carbon dioxide. It's not going to go after the oxygen because it prefers carbon dioxide. This first product, OAA, is going to be converted to a substance called malate, which is then exported to the bundle sheath cells. Malate is going to be converted to pyruvate, and carbon dioxide is going to be released, which is then used in the Calvin cycle. So this figure, 1020, shows the C4 pathway, and this is a good slide to understand to see the differences between the C3 and the C4 pathway. So as you can see, carbon dioxide is entering the mesophyll cells, but again, instead of Rubisco, we have PET carboxylase that's acting as the enzyme. It's going to attach the carbon dioxide or fix it with PEP, and it's going to make oxaloacetate. If you can see in parentheses, it says 4 C's. This means it has 4 carbons. That's why this is called the C4 pathway, the 4-carbon pathway. So you should know this first step so that you can compare it to what's happening in the C3 plants. Eventually, the substances are going to move. Malate is going to break down into pyruvate and carbon dioxide in the bundle sheath cells, and the carbon dioxide is then going to go right into the Calvin cycle, and this will take place in the bundle sheath cells instead of in the mesophyll cells. So this is one adaptation to the C3 pathway. Some plants that use this include sugarcane and corn. So C4 plants Their photosynthesis uses less water and resources than what's used in C3 plants. This is good because suitable agricultural land has been decreasing, partially due to the effects of climate change, but the demand for food continues and is going to increase as the world population increases. Some plants scientists are genetically modifying, so instead of using the C3 pathway, they use the C4 pathway. One plant they've done this with is rice, and they found that compared to C3 rice, C4 rice can yield 30 to 50% more, so that's quite a significant increase. Other alternate pathway is used by plants called cam plants. The plants that do these are called succulents, and this is crassulacean acid metabolism, which is easier to say as CAM. It's abbreviation to fixed carbon. So what CAM plants do is they separate when carbon fixation and the Calvin cycle occurs. So instead of separating where they occur, they're going to separate when they occur. Carbon fixation is going to occur at night and the Calvin cycle during the day. CAM plants open their stomata at night, incorporating carbon dioxide into organic acids that are stored in the vacuoles. Stomata close during the day so they don't lose water, and carbon dioxide is going to be released from organic acids and used in the Calvin cycle. So you see these in plants such as cacti, pineapples, and other types of water-storing plants, your succulents. Figure 1021 is a good comparison slide to help you understand the differences between C4 and CAM plants, the two alternative pathways we talked about. So in C4 plants, it separates carbon fixation and the Calvin cycle spatially. Carbon fixation occurs in the mesophyll cells, and then the Calvin cycle occurs in bundle sheath cells. CAM plants separate the processes temporally, or by the time of day. So at nighttime is when carbon dioxide first enters and is fixed, and during the daytime is when the Calvin cycle occurs. So just as a review of what's going on in photosynthesis, the energy that enters chloroplast as sunlight gets stored as chemical energy in organic compounds. Sugar made in the chloroplasts supplies chemical energy in carbon skeletons to synthesize the organic molecules of cells. Plants store excess sugar as starch in chloroplasts and other structures such as roots, tubers, seeds, and fruits. And just a review of what's happening in the light reactions versus the Calvin cycle. So the light reactions, these are being carried out by molecules in the thylakoid membrane. Light energy is being converted to the chemical energy of ATP and NADPH. Water is being split and oxygen is being released to the atmosphere. The Calvin cycle reactions are taking place in the stroma. The ATP and NADPH that were produced in the light reactions are used to convert carbon dioxide to the sugar G3P. It's then going to return ADP, inorganic phosphate, and NADP plus to the light reaction. So we're going to have the cycle continuing.